All right, so we're going to look at the fundamentals of protein synthesis in this video 7. After this, we'll have two videos really getting more in depth into each individual process, but just a general idea of protein synthesis. So the way DNA is coded, so we've got DNA, which is what we just looked at was a double helix, and it has these base pairs that match up. Each nucleotide will have a base pair on each leg. Now, each group of 3 nucleotides represents an amino acid. And so we're going to look at this molecule called RNA. It's a type of single-stranded molecule that is going to read and make a copy of the DNA in order to take that information out to a ribosome in order to make a protein. Because remember, when we looked at ribosomes in chapter 3, they were the site of protein synthesis. And so we got to get the information from the DNA out to the ribosome, and that's going to have RNA. So each unit of 3 RNA nucleotides represents a genetic code. This sequence of base and the gene determines the amino acid sequence in the polypeptide. Now again, a polypeptide is the name for a protein before it has been completely finished. And a polypeptide just means multiple peptide chains. And remember, going back to again, chapter 2, when we're talking about proteins, that the bond between the 2 amino acids is called a peptide bond. So a polypeptide is just multiple peptide bonds, and that is before the end product, which ends up being a protein. So anyway, each sequence of 3 nucleotides either represents an amino acid, or it could signal the start or stop of the protein synthesis. So we're talking about, remember, the gene is a specific section of the entire genome, but that gene is the information to make one protein. And so we will have little 3 nucleotide sections that are a start and stop area. But in between there is the information. Now, protein synthesis is going to involve 2 steps. One is called transcription. We'll look at that in the next video. And the other one is called translation. And we'll look at that in the video following. Transcription literally means to write something down. So if you took notes while someone's talking, what you were doing is transcribing. You were writing something down. Now, translation is taking something that's been written down, or, well, I'll use that phrase. I mean, it's more than just that, but something that's been written down that is not usable, and translating it and changing it to a usable form. And so we're taking these lists of nucleotides and their bases and turning it into groups of amino acids that will eventually be a protein. So RNA differs from DNA in a couple of ways. The main one, first of all, is that it is a single strand. DNA is a double strand, a double helix. RNA is a single strand. Now again, it has the same thing, sugar, and the sugar of this is ribose instead of deoxyribose in DNA, so it's ribose. But there is also a difference in the bases. While they are almost all the same, RNA uses adenine, guanine, and cytosine, it does not have thymine. Instead, wherever there would be a thymine, it is uracil. All right. So when it goes in and makes a copy, anywhere that the DNA has an A, all right, if we were looking at the other strand of DNA, it would have a T, but since there's no T on we're not on the RNA, when it makes a copy, it puts a U. All right, so instead of T's, there's U, and that's the only difference in the base pairs. So when you're looking at these, remember it has adenine, guanine, cytosine, and uracil. Guanine and cytosine are always the ones that goes together, and uracil and adenine. Now again, if we have a thymine on this side, all right, so this is DNA again. If I have a thymine, it on the other side of it on DNA would be an A. And since this still has A's, the RNA would also be an A, right? So the only change is If there would be a T listed, it is a U, as in uracil. Now there are three main types of RNA. We have messenger RNA. We're going to look at this a lot in transcription and even in translation. We're going to see transfer RNA. This is going to be what does the translating. And then ribosomal RNA, which is all about ribosomes. Ribosomal RNA is the structural component of what makes up a ribosome. So don't overlook the simple fact that ribosomal RNA is about ribosomes. So this is the basic structure of it. And again, this little U, so if it was making a copy over here, what would happen is that would be a T, this would be an A, this would be a C, this would be a C. And so on DNA, boy, I gotta get a pen. When we're making a copy, the T goes to A as always. The A, which usually goes to the T on the other strand of DNA, since we're talking about RNA here, and there's no T, there's a U, right? So hopefully that makes sense. I think that when we went over it in class, everybody seemed to pick that up. It's a little easier to do it on a whiteboard and kind of talking it than this, but I hope that makes sense. We'll see it more again in this, this next video of transcription. Take care and have a great day.